Hello, everyone. What we're going to do in this lecture and in the following one, we're going to be talking about the properties of cell membranes. So what they're made of and how they move material in and out, how is signal transferred through these membranes. What I want to stress on is that what we're going to discuss here is mostly focusing on the plasma membrane, which is the membrane which defines the whole cells on the outside of the cell. However, whatever we discuss here applies also to other membranes as well, such those that you find in eukaryotic cells, forming the organelles and so on. So what we're going to be focusing specifically on this short lecture is the properties of the cell membrane. So what are they made of and how are they fluid? What are the components and so on? One thing, so what we're going to discuss here, this is a quick overview, is first of all, we talk about the structure of the membrane, focusing on what is the meaning when we talk about membrane based on the fluid mosaic model. And then we talk about the different components, which you'll see are lipids, or I should say phospholipids, cholesterol, and proteins. And then we'll talk about how the fluidity of the membrane is affected by the composition in terms of fatty acids and the temperature of the environment. One thing I really want you to take home from this lecture is how membranes are a super dynamic environment. We're used to talk about membranes as these static structures made by phospholipids with some proteins stuck in, other components as well, modify lipids and modify proteins, but in reality, these are super dynamic structures. Think about it. These membranes, they have components that are moving around all the time. The phospholipid and the proteins, they are expanded, they are contracting all the time. They are budding all the time, forming vesicles, or the other way around, they're taking vesicles and fusing with other membranes. So they're super dynamic structures made by different components. In general, functionally speaking, membranes are actually those structures that finally allow for the formation of living organisms. If you think a cell is indeed a unit confined by a plasma membrane, and which, other than defining what a cell is, has a very important role of controlling basically also the environment inside the cells and inside organelles. So they represent a barrier that controls the movement of material coming in and going out. So the nutrients that are needed to be brought in and the waste that needs to be moved out. And we'll talk about that in the next lecture, how this is achieved through diffusion, active transport, and so on. But here we focus only on the structure. In general, membranes have the big role of, at least in eukaryotic cells, to compartmentalize the processes. If you think about organs, are nothing else than the membrane-bound area inside the cell, such as lysosomes, for example, where they have a very specific function. Lysosomes have the function of digesting material that is brought in, or organelles and so on. RAF-ER, for example, has the function of modifying proteins and so on. Other than that, also, they represent a great anchoring sites for a series of enzymes or structures that are needed one after the other to perform a certain pathway. Think about, for example, cellular respiration. We'll talk about the electrotransfer chain. Or think about a series of reactions where the product of one reaction becomes the substrate of the next one, and so on. So if you have these enzymes close to each other, you speed up actually the overall process going from A to whatever it is. So talking about composition, you see that most, the biggest components in cell membranes are phospholipids and proteins, which are not always just pure phospholipid or pure proteins, but sometimes they're modified by the addition of carbohydrates, such as glycoproteins, for example, and glycolipids as well. Now, these two big components, together with cholesterol, at least in animal cells, form a very fluid and mosaic-like structure, which is described by what we call the fluid mosaic model. But it basically entails this, that the membranes are very dynamic structures where all the components, so proteins and lipids, whether they're modified or not, are moving around all the time. And not only that, but being a composition of proteins and phospholipids, they represent a mosaic of these components. In terms of fluidity, as I say, what fluidity means is that the components that are present in the cell membranes, they're not really static, okay, but they're moving around all the time. This slide here refers to the movement of the phospholipids, which can move in very different ways. They can vibrate. So imagine these tails, for example, here, vibrating. They're vibrating all the time. There is energy there, lots of energy. Not only that, but they're moving around. Imagine like they're dancing. These two are going around each other and then floating away in this very, very fluid environment there. They're spinning and so on. So here they do it all the time. Look at this lateral movement. So these two phospholipids, for example, is changing their position, takes place like 10 to the 7 times per second. So extremely, extremely frequent. Other type of movements are less frequent. Like sometimes you might have a phospholipid moving from one layer to the other one. However, you can imagine that this polar has to go through this non-polar environment here. And therefore, this is not a very, very thermodynamically favored type of movement. In fact, it happens about once per month. So it's very, very rare type of movements. So, but you can see, phospholipids are vibrating all the time, moving around, moving sideways, exchanging places, and so on. So all these different type of movements are taking place all the time. And the same thing happens to proteins as well. However, the movement of proteins tend to be less frequent compared to the one of the lipids. And the reason being that the proteins, oftentimes they're anchored to that structure inside the cells referred to as the cytoskeleton. We quickly talked about that in the cell overview, previous lecture on the cell overview. So being attached to this obviously limits the movement in the cell membrane, but it can still move around. How can you visualize this fluidity of cell membrane? Well, you can do this experiment like the one that was done by Fryen and Edidens originally that demonstrated the fluidity of the cells, where you can take two cell types. So for example, in this case, this is a human cell and this is a mouse cell, and you can label. You can do it in different ways using fluorescent dyes, fluorescent antibodies. Doesn't really matter in this case, but you can label the proteins in the membrane with different colors. So now you have that all the proteins in the human cells are red and those in the mouse cells are green. And then you can actually induce these cells to fuse with each other. You can do that in different ways with very high pressure, or you can do that by using viruses. There are different ways that you can force that. Now, you can imagine that as soon as you fuse this, imagine like two soap bubbles fused together, you would have that originally all the proteins in the human cell will be on one side, on one pole, one half, I should say, and the proteins coming from the mouse will be on the other half. Now, if the membrane was not fluid, this will be the permanent situation. However, what Fryen and Edidens observed is that just a few minutes later, so 40 minutes later, you actually have that the two colors, so the proteins coming from the human cell and the one coming from the mouse cell, they actually intermingle with each other. So they mix with each other, suggesting that the proteins, and we know also the phospholipids now, they can actually move around. So these are very fluid structures like that. One important thing I want to throw in here, which connects with what we'll discuss at the end of the lecture, is that if you do the same experiment at lower temperature, you see that this time will actually get bigger and bigger. So this process, this fluidity is affected by temperature, is slowed down by temperature, to a point where you can even stop it if you bring it to extreme low temperatures. Let's talk now about the composition. We talked about how proteins and phospholipids are the main components. Now, depending on the membrane you're looking at, and I'm comparing membranes between different cell types, could be a bacterium or eukaryotic cells, or even within the same cell type, your own cells. take one organelle or another one, the composition or the percentage of the two components is different. Here you see an example. So in plasma membranes in general, the amount of proteins and lipids, they tend to be fairly equal, almost 50-50. There is something missing there. 1% could be cholesterol and so on. Now, if you instead start looking inside the organelles, your mitochondria, you take the inner membrane, which is where cellular respiration is taking place. So there is a very high protein component compared to the plasma membrane, much higher. You have about 75% of the are proteins and 25% are lipids. So you can see how these percentages switch, and it reflects the function of the inner mitochondrial membrane, where you have all these components of cellular respiration, or the last phase, the oxidative process of cellular respiration, confined there. Now, the opposite happens if you look at the myelin membrane. Myelin membranes, you can see in this picture here, are those membranes here that do surround axons in neurons. We'll talk about that in BIO359, and have the function ultimately to limit the loss of ions during the signal transduction in the axons. So they basically work like insulators. Imagine your electrical wires at home. They have this plastic, PVC, I'm not sure what it is, around that it's insulating the actual copper wire inside. Same thing here. This will be the copper wire. The myelin sheets, what they do, they insulate the copper wire, the axon here. And so lipids are much more efficient at insulating than proteins, and therefore this membrane have a very high content of lipid, about 82% versus 18% of proteins. So the composition of the two components is different depending on the membrane you're looking at. Now, the composition is different also if you look at the two layers in the phospholipid bilayer. This is not the same. So the proteins you can find on one side may not be present on the other side. So there is an asymmetry between the two layers. And you can visualize this doing an experiment like this one, where you can actually separate the two layers by first freezing the cells. So imagine you take some cells, you freeze them down very quickly. You can do that very quickly on liquid nitrogen. Once they're frozen, now you can apply some force using like a scalpel type of structure, like a knife edge here. When you do that, what you're doing, you're applying a force concentrated to the edge of the scalpel here, which then will spread through the ice. And the way it spreads is through the areas where there is the least amount of energy required to break up. It's similar to what happens if you think in concrete, you know, on the sidewalks. If you look, when they build sidewalks, every 4 feet, they put some sort of depression, right? And the point of doing that is that if there is any force coming from underneath the sidewalk, what will happen is that this force then will spread to the concrete and will cause a breakage in the areas where you require less energy. Basically, you are directing the breaking of the concrete in specific areas by making them less resistant to force, right? That's hence the depression every 4 feet. So that you still have a functional overall sidewalks, even if you have cracks, but you still have these 4 feet slabs where you can still walk instead of having it all crumbling up. The same thing here. So if you have frozen your cells very quickly and you apply this force, now this force will move through the ice to the areas of where there is less energy required to open up. And where is this area? Is actually that space in between the two phospholipid layers here. So this energy will spread through here, causing the separation of the two layers. Now you can actually take then these two layers and look under the electron microscope, which I'm sure you have in your basement. Now, if you had an electron microscope and you look at that and you look at one side, imagine this one right here, and the other one, you will see that they are different, morphologically different. Just by looking at that, you see the difference. These bumps here are basically proteins embedded in the phospholipid bilayer, in the phospholipid. And so you can see they're much more frequent, for example, in this case, in the membrane interior. So in the inner part of the membrane, this one. Whereas on the outer layer, there are less proteins, clearly showing how the membrane is asymmetric. Talking about components here, so phospholipids are the main lipid that is found in the cell membrane. You see later on, cholesterol is the other one that's very important, at least in animal cells. Now, we already talked about phospholipid, but just reviewing the structure. If you remember, they're made by a molecule of glycerol, which is esterified to two fatty acids, And then the third hydroxy group in glycerol is combined with a phosphate group, which then is attached to a variable group. Okay, so this is just one example of a phospholipid. Now, because of that, these molecules are amphipathic, so they have a clear polar head and a clear non-polar or hydrophobic tail here. And therefore, because of they tend to clump together with the heads connected with each other and with the hydrophobic tails connected with each other, forming the phospholipid layer. Now, depending on the concentration that you have and basically the experimental conditions, you can actually get different type of structures. So if you put phospholipid in water, they could form micelles, a low concentration usually, or liposomes, or if you have two surfaces on the side, you can actually direct the formation of a phospholipid bilayer, like in this case. You can see how a liposome will represent some sort of structures, basically is a spheric form of a phospholipid bilayer, similar to what you find in the cell, imagine in larger dimension. And phospholipid, they do that on their own. They don't require any energy to do that. They do it spontaneously, right? So when they form that, the reason why they form that is basically they put all the polar heads together on one side and on the other side, and all the non-polar tails in the space in between. By doing that, they basically can separate the non-polar hydrophobic component from the hydrophilic aqueous solution on one side and on the other side. They exclude the water, or in this case on the inside and the outside here. By doing that, they create a very stable, thermodynamically stable structure, hydrophobic structure in between. Now, the composition of the phospholipids has a big impact on the fluidity of the deriving membrane. If you remember, we talk about that, how membranes are fluid, how the phospholipids move around and the proteins as well. This is affected by the type of phospholipids that you have here on the phospholipid, making the membrane here. So now, if you remember, when we talk about fatty acid, we talked about how saturated fatty acid, like the two you see in this picture here, they are linear in structure, so they tend to compact very well with each other. They have a higher surface, if you want, where they can contact each other. Whereas when you have some cis-unsaturated, may have trans-unsaturated, but trans-unsaturated, they're not natural. Okay, they're introduced by industrial processes. So I focus on cis-unsaturated, like this one here. You have that the fatty acid will actually kink, will actually turn, and this basically reduce the surface, the contact surface between fatty acids, and therefore, if you want, create a structure which is a little bit more loose. So the consequence of that is that the more fatty acid, unsaturated, cis-unsaturated fatty acids that you have, the more fluid this membrane will become. There's less of chance of these fatty acids to compact, and therefore less forces keeping them close to each other. They have more freedom to move. There is more space here to move. You see how cholesterol somehow compensates for that. And so this creates much more fluid membrane. Now, the type of fatty acid is not the only components that affect the fluidity of cell membrane. The second component is temperature. You can imagine how basically the movement of these phospholipids compared to each other, hence the fluidity, it's also affected by how much energy is present there. So the more energy you have there, the more is the capacity of these fatty acids to separate. They would have enough energy to break those hydrophobic bonds and move around. So at higher temperature, that's where you have a higher amount of energy, membrane would tend to be more viscous. You have more capacity of the phospholipid to move around because there is more energy, and therefore the membrane will be more viscous. Whereas if you are going to lower temperature, now the fluidity of a cell membrane will become less and less. These bonds will become more and more permanent when at lower temperature, to a point that actually the membrane will become some sort of gel, okay, some semi-solid gel. But basically, the fluidity almost will disappear at certain. So the higher the temperature, the more fluid is the membrane, to a point that it can become completely disordered. Lower temperature, more compact, less fluid is the membrane. Now, this is a big problem. Why? We are exposed to high temperature. Well, as human beings, we can compensate having closed environment, heated environments. We have clothes and so on. But imagine animals. Some animals are exposed to fluctuating temperature, very hot or very cold, and so on. So, and obviously the cells that they have in their body, they have to be able somehow to control the fluidity of their membrane. They can't have this fluidity changing all the time. That would affect the movement of material in and out, and so on, the properties overall of the membranes. How do they do that? Well, they do that by expressing some sort of enzymes called desaturases. Now, these are enzymes whose expression, so they're made basically when the temperature drops down. Now, these graphs here shows you how is the expression of these enzymes, desaturases, at different temperature. So at hot temperature, there is very little amount expressed. But when the temperature drops, the amount of this enzyme expressed is much higher. Why? Well, if you think, the lower the temperature, the less fluid the membrane becomes, right? And so how can you compensate that? Is by using the second component that affects the fluidity. So if temperature goes down, now you can play with the composition of the fatty acids. And that's exactly what desaturases do. What they do when they are expressed, they actually convert saturated fatty acids into unsaturated ones. So in the same phospholipid that are present there, they start producing more unsaturated fatty acids, cis unsaturated. Therefore, neutralizing, if you want, the loss of fluidity caused by the lowering of the temperature. So they compensate by that. Now, when the temperature goes up again, these enzymes are not expressed anymore. You can go back to the original percentage, if you want, of unsaturated fatty acid that you had at higher temperatures. So by doing that, the fluidity of the cell membrane can be kept pretty constant at different ranges of temperatures. The last component that we talk about in terms of composition of cell membranes are sterols. And these are key components, again, that affect the fluidity as well, at least in animal cells, where you can find this particular type of sterols, which is cholesterol. If you remember, cholesterol, like phospholipids, is an amphipathic molecule. It has a polar head, which corresponds to the hydroxy group attached to the carbon 3 here in this ring, will be right here. And the remaining part is basically a huge hydrocarbon in a cyclic form, plus there is another component here sticking out along hydrocarbon here too. So because of that, now it can embed in phospholipid bilayers with the polar head, this hydroxy group, together with the polar heads of the phospholipids, and the non-polar component here, the hydrophobic component, embedded within the fatty acid of the phospholipids there. Now, why is cholesterol essential and found in animal cells? Is because it can help actually compensate the fluidity. And it can help compensating the fluidity both at high temperature and at low temperature. Now, at high temperature, if you remember, there is a lot of energy. The membrane would tend, or the phospholipids in the membrane would tend to move around quite a bit. By doing that, the fluidity will be very high. Now, if you have molecule like cholesterol, can basically fill some of those gaps left by cis unsaturated fatty acid. By doing that, they increase the surface, the contact surface here within this layer, and therefore reduce the fluidity at high temperature. Now, the same thing happens when the temperature goes down. The temperature goes down, the fatty acid would tend to actually compact better. But now having cholesterol in there, it actually prevents the fully compacting of particularly saturated fatty acid. You have something there that creates a gap. If these are saturated fatty acids, creates this gap and therefore reduces the overall capacity of compacting and reducing the fluidity. So that by doing that, it still keeps the fluidity to a certain level, even at low temperatures. Last component we'll talk about here is membrane protein. So in general, there are two types. We'll talk about them in the next lecture. So we don't spend a lot of time here, but membranes, proteins in the membranes can be divided into two groups if you focus on their location, or four main groups if you talk about their function. If you talk about their location, they can be referred as integral or peripheral proteins. Integral proteins are proteins that do enter the phospholipid bilayer, but either going through the whole phospholipid bilayer, like this or this protein here, or even just one of the layers, but still enter the hydrophobic component. Or there are peripheral proteins which are associated with the cell membrane weakly. So they're not entering the cell membrane, but they're attached to either the phospholipids here or other proteins that are present, or integral proteins that are present in the cell membrane. Functionally, instead, the type of proteins that you can find in the cell membrane can have four different functions. They can be involved in the movement of material in and out, so they're called transport proteins. We'll talk about that in the next lecture. Or they can be involved in attachment or recognitions of the cells. You see here some examples, cell recognition molecules here, which allow two cells to actually know who they are by connecting these two proteins, or they can transfer signal. We'll talk about that in the next lecture. They can have also enzymatic activity. So there are different ways in which proteins in the membrane can be classified, either by their location or by their functional types. Now, proteins that are integral, so they have to go through the phospholipid bilayer. And in order to do that, they have to have specific properties which allow them to embed within a hydrophobic environment, like the phospholipid bilayer. And of course, they have to have also properties that allows them to be in the aqueous environment, either outside or inside or on both sides of the cell membrane. And so membrane proteins, integral membrane proteins, they would have a common property of having what is called a transmembrane domain, which consists of 17, 20 nonpolar amino acids that can span, they are nonpolar, so they all have nonpolar R groups sticking outside. Usually they take the conformation of an alpha helix. These tubes you see here, they're alpha helixes, and they would have the R groups projected outside here. So all the nonpolar R groups will be projected outside in the phospholipid bilayer, allowing basically these protein to stay inside there stably. Now they can cross the membrane once or multiple times, like this protein right here. And together with that, they would have also other domains on the outside and on the inside, which instead, being on an aqueous environment, would have polar amino acids sticking out there. You see how these domains can have different function. When we talk about in the next lecture about signaling across the membrane, you see that receptors do have a signal recognition domain here, polar 1, then they would have a transmembrane domain, and then they would have a enzymatic domain, a kinase domain on the inner side. So they can have different domains present in there. When it comes to instead peripheral proteins, if you remember, these are proteins that are weakly attached to the cell membrane. They can actually fairly easily be washed away using detergent or high ionic concentration and so on. So, and the reason being that they're not really embedded inside the phospholipid bilayer, but they're attached to components of the phospholipid bilayer, like phospholipids or proteins. And they do that through non-covalent bonds, so weak bonds like hydrogen bonds and ionic bonds. And so they can be obviously composed of different polar, non-polar amino acid, but usually with the polar amino acid on the outside, so they can still stay in the aqueous environment. In summary, finishing up this lecture, what did we talk about here? We talked about the structure of the cell membrane. First, we focus on the fluid mosaic model, discussing how membranes are fluid, how phospholipids can move around, how proteins, particularly phospholipids, how they can move around in the cell membrane, making it fluid. Then we talk about the asymmetry between the phospholipid bilayers as well. And then we talk about the composition of the fatty acid and how fatty acid and temperature and cholesterol ultimately they affect the fluidity. Finally, we talk about the composition in terms of phospholipid, proteins, and cholesterols, and we focus on how proteins can be classified, the proteins found in the cell membrane, based on their location or their function in the membrane.